Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine if we could tune our cancer-fighting T cells into, like, surgical strikes, delivering potent therapies only inside a tumor, leaving healthy tissues completely untouched. What's truly remarkable here, and what we're diving into with a groundbreaking Nature paper from 2025, is how researchers have learned to rewire CRT cells using their own genetic code to do just that.
Yeah, and it's not about adding some new synthetic switch.
Exactly. It's about tapping into the cell's native logic for precision delivery. It feels like a huge leap, especially for treating solid tumors. CRT cell therapy has been just incredible for blood cancers, really offered new hope.
Oh, absolutely. A game changer.
But, well, solid tumors are just a different beast, aren't they? Much tougher.
That's right. Yeah, if we connect this to the bigger picture, CRT cells have revolutionized blood cancers, sure. But solid tumors, their effectiveness is often limited. You've got this hostile tumor microenvironment suppressing the immune response, and then there's antigen heterogeneity.
Meaning the tumor cells don't all look the same to the T cells.
Exactly. So researchers tried armoring the KRT cells, basically engineering them to secrete powerful stuff like IL-12, IL-18, checkpoint traps, things to help them fight better.
Okay.
And these showed improved efficacy in, early mouse models, seemed promising. But then when these armored cells went into first-in-human trials, things got complicated. They often ran into severe systemic toxicity.
So the weapons were firing everywhere.
Pretty much. These powerful transgenes would activate outside the tumor, causing widespread inflammation in healthy tissues. It was like ordering a blanket bombardment when you just needed a precision strike. Not good.
So after those early setbacks, how did they pivot? What was the next step for a safer, more effective strategy?
That's exactly where this paper comes in. They focus right on that critical unmet need.
Yeah?
How do you get these potent payloads to fire only inside the tumor? And importantly, without complex external drug switches or really burdensome manufacturing? The elegant solution they explored was plugging directly into the cell's own endogenous gene control systems, essentially rewriting the delivery rules using the genome's native logic.
Using what's already there.
Precisely. So the therapeutic payload only gets expressed where it should be in the tumor microenvironment, where native signals like T-cell activation or, say, hypoxia naturally go up. More potent, but also, crucially, far safer for solid tumors.
Okay, so the first hurdle was just proving they could even do this, right? Hooking a payload onto the T-cell's own genetic controls using CRISPR. They started with a gene they knew well, PDCD1, to see if they could really rewire its expression.
Yeah, that was the proof of concept.
They developed a CRISPR knock-in strategy, using CRISPR to slot new genetic material into a specific spot in the cell's DNA. They aimed for these native genetic sites, endogenous loci, and they picked PDCD1, the gene for PD1.
Right, because PD1 expression ramps up a lot when T cells get activated, like when they encounter a tumor. It's a known activation marker.
So they designed this homologous repair template, like a specific blueprint.
Exactly, a blueprint to insert a reporter gene, GFP, that's the one that glows green.
Right, so you can easily see where the gene's active.
Yeah, they put GFP right after the PDCD1 start codon. Then they worked out a protocol to get high-efficiency editing in primary mouse T cells.
And did it work?
It did. When these modified OTI cells, those are T cells recognizing a specific antigen, were engineered to express GFP under the PDCD1 promoter, and then stimulated in the lab with activating antibodies or tumor cells, they successfully ramped up GFP expression.
Okay, so that was the first big sign they could hijack an internal switch.
It was a critical first piece of evidence, much more nuanced than just adding a new kind of blunt synthetic switch, a key step towards making these CRT cells smarter.
Right. So the next step was moving into a living system, into mice.
Yeah.
They put these engineered T cells into mice with AT3 ova tumors. The question was, could PD-CD1 drive expression mainly in the tumor?
It did.
In the mice, the PD-1-GFP OTI cells showed strong GFP inside the tumor, but significantly lower GFP in the spleen, which was their healthy tissue control.
So differential expression in vivo, that's good. Building on that, they tested the therapeutic potential and, importantly, safety. They hooked up various pro-inflammatory cytokines to this PD-CD1 switch.
These are the signaling molecules that can be really toxic systemically.
Exactly. So for things like TNF, IFNO, even IL-2, these PD-1 rewired T cells actually showed significantly enhanced tumor control and, importantly, without obvious toxicity.
That sounds really promising.
It was very encouraging, yeah.
But then came IL-12.
Yes, IL-12, that's where the plot twist happened. When they assessed the PD-1 IL-12 OTI cells delivering that really potent cytokine, the results were dramatic.
Not in a good way.
Not at all. Severe systemic toxicities. The mice lost weight rapidly, had huge increases in IL-12 levels in their blood everywhere. They eventually had to be ethically euthanized.
Oh, wow. So that elegant theory hit a hard reality check.
It really did. It was a stark reminder. And the immediate question became, why? Why did PD-CD1 seem okay for some cytokines, but was catastrophic for IL-12?
Right.
It just highlighted that while PD-CD1 offered some tumor specificity, it just wasn't stringent enough. It wasn't tight enough.
Still too much leakage into the rest of the body.
Exactly. It resulted in unacceptably high levels of peripheral expression for a super toxic payload like IL-12. They realized, okay, PD-CD1 isn't the answer for everything. We need alternative promoters with even stricter, more tumor-locked expression patterns.
Okay, so PD-CD1 wasn't the smart bomb switch they needed, especially for the really potent stuff. They needed better delivery rules written right into the genome, something super specific. So how do you find those?
Well, they cast a wide net. They did RNA sequencing measuring gene activity on CRD cells, pulled from both tumors and spleens.
In both mouse models and human tumor models.
Yes, exactly. They were looking for genes that were really cranked up, upregulated only in the CRT cells inside the tumor compared to those same cells in the spleen, the healthy tissue proxy.
Smart. Comparing the on-state in the tumor to the off-state elsewhere.
Precisely. By correlating all that data, they narrowed it down to 27 promising candidate genes that showed this high differential expression.
27. Okay, so then they had to screen those.
Meticulously. They started knocking out each of those 27 genes in human CRRT cells.
Why knock them out?
To check the impact on basic T cell function, making cytokines, killing tumor cells, proliferating, they had to make sure disrupting the gene didn't accidentally cripple the T cell itself.
Ah, right. Don't want to break the delivery truck while trying to improve the delivery instruction.
Exactly. So genes like AK4 and FOSB got kicked out early because knocking them out reduced cytokine production. Not good.
Okay.
Then more in vitro testing. They inserted GFP into the top shortlisted genes, stimulated the cells with tumor cells, and they found that NR4A2, PD-CD1 as a control, RGS16, and RGS2 led to significant GFP induction when the T cells saw tumor cells.
And others got excluded.
Yeah, DUSP4 and TNFA-IP3 were out because they showed high baseline GFP even without stimulation. That's a red flag for potential toxicity in vivo.
Right. You want the switch to be firmly off until it sees the tumor.
Exactly. So the real test, the definitive one, was in vivo. Back to the tumor-bearing mice.
Checking those remaining candidates for truly strict tumor-only GFP expression.
Yes. And this is where the champions emerged.
We're the winners.
Well, while all the tested promoters showed some increase in GFP inside the tumor compared to the spleen, a few stood out. CLU-GFP, NR4A2-GFP, and RGS2-GFP SAR T cells showed remarkably stringent tumor-restricted expression, like less than 10% GFP-positive cells in the spleen. Very tight control.
And among those,
NR4A2 really rose to the top. It gave the highest GFP expression inside the tumor while keeping expression minimal in the liver, another key organ for toxicity checks. That made it look ideal for highly toxic payloads, like the problematic IL-12.
okay, so NR4A2 for the really potent, need to be super careful stuff. What about RGS16?
RGS16 was interesting. RGS16 GFP-CHAR T cells showed similar spleen expression to PD-CD1, so maybe slightly leakier than NR4A2. But crucially, they expressed significantly higher levels of GFP within the tumor itself. So more firepower inside the target zone.
Exactly. That suggested RGS16 might be perfect for transgenes that need really strong intratumoral expression, maybe for payloads with a milder toxicity profile, where you have a bit more wiggle room.
Got it. So they didn't just find one switch, they found potentially two. tailored for different jobs, NR4A2 for tight control, RGS16 for high expression.
Precisely. A toolkit.
Okay, so armed with NR4A2, they went back to IL-12, right? The cytokine that caused all those problems with PD-CD1. Could NR4A2 finally tame it?
That was the million-dollar question, and the results were, well, striking.
How so?
NR4A2 GFP OTI cells not only showed significantly more GFP upon stimulation, but critically, they had far more tightly regulated GFP expression in vivo compared to the original PD-1 GFP cells.
How tight are we talking?
Less than 1% of the OTI cells express GFP in the spleen. That's a massive improvement in safety right there.
Wow, okay. And they also compared this to the standard synthetic approach, the NFAT promoter.
They did, and that comparison really drives home the advantage of the endogenous approach. What did they see?
Well, synthetic NFA also limited GFP before activation, which is good. But in vivo, those NFA-GFP CAR T cells still showed noticeable GFP, maybe around 10% in lots of peripheral organs. Spleen, blood, liver, lung, even brain and kidney.
So still some off-target activity.
Right. But in stark contrast, the NR4A2 GFP, say CAR T cells, they maintained a maximum of just 2% GFP expression across all those peripheral sites tested.
From 10% down to 2%, that's a big difference when you're talking about something toxic like IL-12.
Absolutely paramount for safety. That difference in stringency is huge.
So, okay, NR4A2 provides this super tight control. Did it translate to better outcomes with IL-12? What happened to the tumors this time?
The results were clear and definitive. NR4A2 IL-12 OTI cells showed significantly enhanced anti-tumor efficacy. The mice survived longer, and this effect held up even when they used lower doses of the T cells.
And the toxicity.
This is the crucial part. In direct contrast to the disaster with PD-1 IL-12 cells, no toxicity was observed in mice treated with the NR4A2 IL-12 OTI cells. None.
That's incredible. Potency without the poison.
Exactly.
And how did it stack up against the synthetic NFA promoter carrying IL-12?
Same story, essentially. NR4A2 IL-12 murine CAR T cells showed significantly better therapeutic effects, better survival, worked against tough tumor models, and again, no observable toxicities. The NFA IL-12 CAR T cells, on the other hand, severe toxicities, rapid weight loss, other clinical signs, it just wasn't safe.
So the endogenous approach wasn't just a little better, it was qualitatively different. It achieved what the others couldn't. Powerful therapy without the dangerous side effects.
Precisely. A major step forward.
Now, you mentioned earlier that these things might also wake up the patient's own immune system. It's not just the CRT cells doing all the work.
That's another fascinating layer to this, yes.
What did they find about how the NR4A2 IL-12 T cells behaved that suggested this broader effect?
Well, first, the NR4A2 IL-12 OTI cells themselves seemed more active. When stimulated, they pumped out more IFN and TNF potent anti-tumor cytokines, and they showed higher levels of Ki67.
Which means they're proliferating more, growing faster.
Exactly. Indicates a more activated, robustly dividing state right there in the tumor. So they're not just delivering IL-12, they seem to be functioning better as T cells too.
And this enhanced function it led to epitope spreading. Can you unpack that a bit? It sounds important for long-term cancer control.
It's absolutely critical, especially for solid tumors, which can be very heterogeneous. Think of it like this. The CRT cells initially attack cells showing the target antigen, let's say antigen A.
Okay.
As those tumor cells die, they break apart and release all sorts of other tumor-specific molecules, maybe antigen B, antigen C, etc., that weren't visible before.
Spilling their guts, basically.
Right. And the IL-12 being produced locally by the CRT cells helps activate the host's own immune cells, like dendritic cells, which pick up these newly released antigens B and C. Those dendritic cells then travel to the lymph nodes and essentially teach the host's own T cells to recognize and attack antigens B and C.
So the initial targeted attack spreads the word, teaching the rest of the immune system to fight a broader range of tumor targets.
Precisely. It broadens the immune response beyond the single engineered target.
How did they show this was happening?
They used those congenic mice again, allowing them to distinguish donor CAR T cells from the host's own T cells. They saw more host CD8 plus T cells specific for the original target antigen over in the spleens of mice treated with NR4A2 IL-12 cells. And even more importantly, they found significantly more host CD plus T cells producing IFN-FN and TNF right inside the tumors and in the draining lymph nodes, plus more proliferating Ki67 plus host T cells.
So the host's army was getting activated and joining the fight.
Definitely. And TCR sequencing, looking at the specific T cell receptors in the lymph nodes, showed expansion of multiple new host T cell clonotypes. Clear evidence of an endogenous response being stimulated beyond the initial target.
And did this lead to longer-term protection?
It did. Mice that had been cured by the NR4A2-IL12 CRT cells were significantly protected when they were rechallenged later with the original tumor type, even without the CIR target. This points to durable, broader immunity.
That's incredibly powerful. A precision strike that then calls in the whole army and trains it to recognize more targets. Huge implications for dealing with diverse, evolving tumors in people.
Yeah, absolutely.
Okay, this looks fantastic in mice, but the big question is always, what about humans? Clinical potential.
Right, the translation step is key. And they did extend these findings into human tumor models.
Using human CAR T cells.
Yes, they made human anti-LAY CAR T cells targeting that LAY antigen found on some human cancers with the NR4A2-IL12 knock-in. These cells expanded well, produced more IL-12, IF-9, TNF, and IL-2 in lab tests, and showed significantly better anti-tumor responses in mice bearing human OVCR3 ovarian tumors.
And crucially,
No observable toxicities. Again, a stark contrast to PD-1-IL-12 CAR T cells, which were toxic in the human cell setting, too.
they even looked at cells from actual patients, right?
Yeah.
Not just healthy donors.
Yes, that's a really important validation. They took T cells from patients with DLBCL, that's diffuse large B-cell lymphoma and multiple myeloma. They engineered these patient-derived cells with either the NR4A2-GFP or RGS16-GFP constructs.
And they behaved similarly.
Very similar results to the healthy donor cells. Stringent, inducible GFP from NR4A2, potent GFP expression from RGS16. It suggests the approach works with patient cells, too. And they even looked at publicly available single-cell RNA sequencing data from human tumors.
What did that show?
It confirmed that you find higher frequencies of T cells naturally expressing NR4A2 and RGS16 inside various human tumors compared to in the peripheral blood. This supports the idea that these switches are indeed active in the right place in humans, suggesting broad applicability.
That's really encouraging. So if NR4A2 is the go-to for potent things like IL-12, what about RGS16? Can you really mix and match promoters based on the payload?
It seems so. This design flexibility is a major plus. They tested this with IL-2, a cytokine with a milder toxicity profile than IL-12. NR4A2-IL2 CAR T cells only gave a mild therapeutic boost. But RGS16-IL2 CAR T cells, leveraging that promoter's ability to drive higher expression inside the tumor, showed significantly higher IL-2 levels and much better anti-tumor effects.
And still safe.
Still safe, no toxicity observed. It demonstrates that RGS16 is a great choice for driving strong intratumoral expression of factors where you might tolerate or even want higher local levels, perhaps because the factor itself is less systemically toxic.
So you can tailor the promoter choice to the specific transgene and its properties.
Yeah.
That's a sophisticated level of control.
It really is a customizable approach.
What about the practical side? Making these cells for potential clinical use, manufacturing is always a bottleneck.
They addressed that too. They showed they could do a one-step manufacturing protocol. Meaning they could simultaneously knock in the therapeutic transgene into NR4A2 or RGS16 and knock in the CAR-R into the TUR-AC locus, a standard safe spot all in one go.
That could seriously streamline production, make it faster, maybe cheaper.
Potentially, yes. Big advantage for clinical translation. And one more thing, the system seemed pretty sensitive. Sensitive how?
It worked effectively even against tumor cells with low levels of the target antigen, or when the T-cell receptor affinity wasn't optimal. These are common challenges in the clinic, so showing robustness there is important.
Okay, so pulling back a bit, thinking about the whole study, what's the real core innovation here? It feels like more than just finding a couple of good promoters.
You're right, it is more fundamental than that. The core innovation is repurposing the cell's own endogenous gene regulatory machinery to express these payloads. Unlike synthetic promoters, which are often just short snippets of DNA trying to mimic regulation, this CRISPR knock-in taps into the full regulatory complexity of the native gene locus.
Including all the enhancers and other control elements nearby.
Exactly. All the intricate cis and trans-regulatory elements that nature evolved over millennia to control that gene's expression precisely in space and time. This gives you much more robust, nuanced, and physiologically relevant control. It's like writing a built-in safety directly into the cell's operating system. So it's not just on or off, it's tuned. And it switches off properly when needed.
Yes, that's a key advantage over many synthetic systems, which can sometimes be leaky or stay on too long. This endogenous control offers a level of sophistication that's hard to replicate artificially.
So what does this shift mean for the future? How might this inspire other researchers? Where does this lead?
Well, it provides a kind of general recipe, doesn't it? A template for achieving tumor-selective potency that could be plugged into many existing CAR T-cell pipelines. The benefits are clear: lower systemic toxicity risk, better signal-to-toxicity ratio. It directly tackles key solid tumor challenges like immunosuppression and heterogeneity by engaging the host immune system via epitope spreading.
And it seems versatile.
Immensely versatile. You could use this CRISPR knock-in approach to express almost any DNA-encoded factor in a tumor-restricted way. Future studies could look at safely delivering other potent factors, maybe IL-15, which has also had toxicity issues.
Or combining things.
Definitely. Multiplex editing. Imagine knocking in a payload and simultaneously knocking out genes that limit T-cell function, maybe even NR4A2 or RGS16 themselves, as they can contribute to T-cell exhaustion later on.
Wow, okay.
Or co-expressing multiple synergistic factors. A multi-pronged attack, all under endogenous control.
And beyond CAR T cells.
Absolutely. This whole concept of endogenous gene rewiring could be applied to other cell therapies, CAR macrophages, SAR NK cells for cancer, or even things like TREG therapy for autoimmune diseases. You just need to identify the right tissue-restricted promoters for those cell types.
Of course, it's not perfect yet, right?
What are the remaining challenges?
No, definitely challenges remain. Choosing the right target antigen is still critical to avoid attacking healthy tissues that might express low levels of it. That on-target off-tumor toxicity is always a concern. The paper highlights good candidates like LAY, STEAP2, GPC3, or neoantigens with minimal expression elsewhere.
And the gene editing itself. Yeah, CRISPR is powerful, but you always need to be vigilant about potential low-frequency off-target edits or chromosomal changes. Rigorous safety testing and validation in human trials are absolutely essential.
So wrapping this up, what's the big takeaway? We've seen how using a cell's own genetic wiring can lead to cancer therapies that are powerful but also incredibly precise. Turning CAR T cells into these smart, context-aware couriers, delivering therapy just where and when it's needed.
Yeah, I think on-site bioreactors with built-in safety is a good way to put it. It marks a real shift from just armoring cells to making them truly intelligent and context-aware. It's a foundational change, offering a template for safer, more effective next-gen treatments.
It really makes you think. So here's a final thought for you. If we can truly reprogram cells to follow these complex native rules for deploying therapies, essentially writing new, precise logic into the genome's own language, what other complex biological challenges, maybe way beyond cancer, could we tackle with this level of control? The potential for truly smart cell therapies feels enormous, maybe even mind-boggling.